Welcome to this e-learning video where I will give a short introduction to the TropFishR package. You will see how to use this package for the estimation of parameters related to growth, mortality and selectivity using length data and an application to mesopelagic fish species. I will start with an introduction of the mesopelagic zone and the motivation of a potential fishery targeting species in this region, after which we will dive into the methods. I'll show you the reasoning and the benefits of using length-based methods for estimating life history parameters, the TropFishR package, its origin, methods and assumptions, and I'll give you some examples on how to use it, including an application of this model to estimate life history parameters for mesopelagic fish. I'll end by showing you some examples of future steps using the TropFishR package to estimate, for example, stock status. This material is produced by the Horizon 2020-MEESO project, which stands for Ecologically and Economically Sustainable Mesopelagic Fisheries. MEESO will determine the potential of sustainably exploiting the mesopelagic biomass for products intended for the human food chain. It focuses on two key mesopelagic species in the North Atlantic region, the deep-sea pearlside and the glacier lanternfish. Now, part of this project aims at establishing new tools for assessment and fisheries management of this potential new fishery. And one of those tools is TropFishR. The mesopelagic layer, or the ocean twilight zone, is a layer of water that stretches around the globe and lies between approximately 200 and 1000 meters depth. And this layer is inhabited by a diversity of species, ranging from crustaceans to cephalopods to, of course, fish. Now, mesopelagic biomass estimates are very high, but also very variable and range between 1 and 10 billion tons globally. Now, the mesopelagic community provides some important ecosystem services. Many species in this layer perform diel vertical migrations, where during the day, individuals stay in deeper and darker waters to hide from visual predators, and during the night they then move to shallower water layers to feed. And with those daily migrations, part of the mesopelagic community has an important role in the sequestration of carbon to the deep ocean. Further, mesopelagics form an important resource for other species such as marine mammals and tuna, or more locally in the North Atlantic for commercially important species such as blue whiting. Today, there is interest to start exploiting species in this layer, because as the human population grows, the demand for food, including marine products, also increases. The Food and Agricultural Organization of the United Nations reported that while 66% of the exploited fish and shellfish stocks are fished in a sustainable way, 34% of those stocks is still fished above biologically sustainable limits. Now, the sustainable exploitation of new marine resources, such as mesopelagic species, could partially relieve the fishing pressure on existing marine resources. The mesopelagic catches will be processed into fish meal and fish oil that, for example, could be used as feed in the increasing aquaculture industry. The two main species of interest for potential exploitation in the North Atlantic region are Malgolicus muelleri or Muller's pearlside and Benthosema glaciale or the glacier lanternfish. Those are the two most abundant species in the region and are interesting target species due to their high fat content. Malgolicus is a small and short-lived species which reaches a maximum age of approximately 3 years and matures already after 1 year. Over its lifespan, it reaches a maximum size of 7 centimeters. The species is distributed globally, but mainly concentrated in the temperate waters of both the Atlantic and the Pacific Ocean. Benthosema then reaches a maximum age of approximately 8 years and a maximum length of approximately 10 centimeters. And it matures only after 2 to 3 years. This species is concentrated mainly in the Atlantic Ocean. The first step towards sustainable harvesting of those mesopelagic species is the estimation of robust growth and mortality estimates that are required in stock assessments and estimations of reference points according to maximum sustainable yield. As shown on the maps before, both species are distributed quite widely, but there are only limited estimates related to growth and mortality available in time and space. In our case, we used TropFishR to analyze the available length-specific information to investigate regional and seasonal differences in growth and mortality, as well as differences in selectivity. We attempted to identify sources of variability in life history characteristics that might inform and influence future stock assessments. We also tried to identify spatiotemporal variability, possibly suggesting different stock units. And our work had the following specific aims. So first, it was to estimate growth parameters and mortality rates and their uncertainties for both species in the North Atlantic region. Then it was to identify spatiotemporal variability and population dynamic parameters, taking into account both horizontal distribution patterns and temporal variability. And then it was to evaluate and discuss the management implications, the potential to manage each of the two species as a single stock, and possible optimization of sampling strategies. Why did we choose a length-based approach? That is because length-based methods allow to take into account the full variability of sizes at different times and places and from different gears. Further, length frequency data is also easy to collect compared to age data and gives the opportunity to set up sampling systems that are statistically cost and labor efficient. Fish lengths are also directly related to vital rates, sexual maturity, and fisheries and gear selectivity. In the case of an unexploited situation, such as for mesopelagic species, there are also challenges in relation to the use of common assessment methods and models. Age-based models can't be used as there is no or very limited time series of age readings available. There is no catch data available to be used in production models, and also abundance estimates from acoustic surveys in the mesopelagic zone are still challenged by the distinction between trophic levels, species, and stocks in this densely populated layer. Length data, however, are routinely measured during surveys targeting also other species and therefore provide a suitable alternative. The TropFishR package is an R package compiling single species stock assessment methods designed specifically for data-limited fish and shellfish stocks using length frequency data. And it is based on the framework and model given by the FAO manual on tropical fish stock assessment. The current version of TropFishR can be downloaded from both CRAN or GitHub, examples you can see here. The TropFishR package is an R package including methods for estimating biological population dynamic parameters, such as individual growth rate with ELEFAN, that can then be used as an input for natural mortality estimations with empirical formula, and total mortality estimations with the length-converted catch curve. TropFishR further includes methods to estimate fishery-related parameters such as exploitation rate and selectivity, and methods to assess stock size and status. Here we will focus on the methods for estimating biological and fishery-related parameters. As a first step, we're interested in estimating growth, as this is information required in all later estimations related to mortality and selectivity, and it is also required in stock assessments and estimation of MSY reference points. The growth model implemented within TropFishR is the von Bertalanffy growth function, which describes the length at age according to the asymptotic length L infinity, the von Bertalanffy growth constant K, and T0 indicating the theoretical age when length equals 0. The figure on the right here shows the basic form of the function under different values of K. To estimate those growth parameters, TropFishR uses electronic length frequency analysis. With this method, the growth of the fish is followed by identifying the mean length of the cohorts over time. Each peak, as seen on the figure, is assumed to indicate the mean length of a cohort or year class. Now, length distributions of fish of a given year class, all of approximately the same age, doesn't all have the same length, but rather follows a normal distribution due to, for example, differences in sampling time or individual growth. Therefore, ELEFAN consists of two steps. First, we restructure the data to identify the peaks or modes in the data. And in a second step, we then fit growth curves through those peaks. The data restructuring works as follows. First, we aggregate the length data into length classes according to a bin size that is as small as possible, but at the same time reduces the noise in the data. Those length classes are then also aggregated and sorted by time, for example by month, quarter or year. And then we do the actual restructuring by choosing an appropriate moving average. As a rule of thumb, this moving average should be approximately equal to the number of adjacent bins in the first cohort. The frequency of each length class is then divided by the moving average in order to identify the final length peaks in the data. Practically, it is recommended to start by visualizing the raw and restructured length frequency data, which helps to determine an appropriate bin size and the moving average. The function LFQ-modify allows to change the bin size, and the function LFQ-restructure is used for the actual restructuring process, where the argument MA allows for the control of the number of bins used to calculate the moving average. It is also possible to apply an additional square root transformation in the restructuring, which reduces the weighting of large individuals. The bottom panel here shows the length data after restructuring with a much clearer identification of the modes. So here the positive peaks are indicated in blue, while the negative peaks are indicated in red. Subsequently, different growth curves are fitted through those modes. Each set of parameters obtains a score according to the number of peaks that are passed through. The best fitting parameter set is judged according to the number of peaks that are touched by the growth curves, according to the ratio of the explained sum of peaks and the available sum of peaks. Different methods to estimate growth are available in TropFishR. We here focus on ELEFAN with a genetic algorithm, or ELEFAN GA. This is a stochastic search algorithm inspired by the basic principles of biological evolution. Here, each iteration, the best parameter combinations, or the combinations with the highest fitness, are then passed on to the next generation or iteration. It is also possible with this method to account for seasonalized variation in growth, which includes two new parameters into the von Bertalanffy growth function. C, indicating the amplitude of the seasonal oscillation, and TS, which indicates the fraction of the year where the sine wave of the oscillation begins. It is recommended to first use the simple growth model and then proceed with a seasonal growth model if there seems to be some systematic offset between the modes and the growth curves. The reasoning for this is that the seasonalized function increases computational effort and that the chances of introducing spurious results increases due to the increased number of estimated parameters. With the ELEFAN GA function, it is necessary to identify the lower and the upper bounds of the optimization search space. In this case, the limits for L-infinity are based on the maximum length in the data. All the other parameters are limited to ranges between 0 and 1. Setting this search space is an important step, and it is recommended to do sensitivity analysis to select appropriate bounds. After running the ELEFAN GA algorithm, it is also recommended to visually inspect the results again. Overall, to estimate growth, there are 4 important steps. First, it is to find the optimal bin size in order to amplify the signal in the data while reducing the noise. Then it is important to test different combinations of bin size and moving averages. And when you're satisfied with the restructured data, it is important to double check the settings of the genetic algorithm. Lastly, it is important to also provide a visual fit of the results as a check. The natural mortality rate M is another influential parameter of stock assessment models and can be caused by different factors, such as predation, starvation, or abiotic stressors. The decline in the number of individuals due to natural mortality is given by a simple negative exponential model, with N0 the number of individuals at the beginning of the time period. When no controlled experiments or tagging data is available, the main approach for its estimation is to use empirical formulas. There are plenty of different empirical formulas for the estimation of this parameter that rely on correlations with life history parameters and/or environmental information. TropFishR has several of those formulas included, and here is one example based upon a meta-analysis of 201 species that require estimates of the von Bertalanffy growth parameters. In TropFishR, M can be simply estimated with the function mEmpirical, with only the estimated growth parameters as an input. The total mortality Z, which is the sum of the natural mortality and the fishing mortality, can be estimated in TropFishR using the length-converted catch curve. The length composition of a population provides information about the mortality rate in a population, but this mortality signal is confounded with information about growth parameters and selectivity. Therefore, this method requires as input not only length frequency data by year, but also von Bertalanffy growth parameters. The method assumes also a steady state. The way it works is that the natural logarithm of the numbers in each length group are plotted against the relative age, and this number is then divided by the time to grow through the length class. This gives a curve according to the following figure, where the slope of the curve on the right-hand side corresponds to the total mortality. The catch curve method also allows for the estimations of selectivity parameters, which can be deducted from the left-hand side of the curve. This is under the assumption that if Z were to be constant over the considered size range, this extrapolation represents the number that would have been caught if there was no exploitation. Therefore, the estimate of selectivity is given by the ratio between the observed frequency and that of the back interpolation. It is currently only possible to use a logistic selection ogive in TropFishR typical for trawl net selectivity. The total mortality rate is estimated with a sample of the catch, representative for the whole year. Besides changing the bin size, the function LFQModify allows to rearrange the catch matrix into this required input format and also for optional pooling of the largest length classes into a plus group. The catch curve can then be applied to the vectorized dataset. It is possible to use either an interactive plotting function, where the user selects points to be used in the regression analysis by clicking on the first and the last point to be included, or to define the range using the regint argument. The uncertainty of the growth parameters can be estimated with a non-parametric bootstrapping approach, where 500 random subsamples of the length frequency data are taken, and for each subset the growth parameters and mortality parameters are estimated. This allows for the estimation of maximum density estimates and of confidence intervals of all parameters. Now this method is still under development, but can be already downloaded through GitHub. In our mesopelagic use case, we analyzed the length data of the two species in Norway, in Iceland, and in the Bay of Biscay. We aggregated the length frequency data by sea region as the fish were sampled in all regions with different gears and mesh sizes and according to different sampling frequencies. In a first step, we standardized the data. So we converted some samples from total length into standard length. We raised the frequencies to the entire catch if subsamples were measured, and we removed outliers. Here on the right, you see the length frequencies from Maugolicus in the Bay of Biscay, and as you can see, only 2 peaks can be identified, and the data is quite a bit noisier than in previous examples. Then we did a sensitivity analysis for each combination of bin and MA to select the appropriate restructuring parameters. We finally selected a bin size of 1 and a moving average of 9, as it allowed for the highest detail while at the same time reducing the noise in the data. The figure here shows growth parameters estimated for each different combination of restructuring parameters that we tested. And as you can see, it is really important to be careful with the restructuring, as it can give quite different results. Next, we also did a sensitivity study on the model settings. Influential parameters were the bootstrapping search space, the maximum age, or the number of peaks fitted, the runtime of the optimization, and the number of permutations of the optimization algorithm. These settings were therefore also selected carefully. Limiting the search space of k between 0 and 2, for example, as you can see in the bottom row, leads to quite different estimates of L-infinity and T-anchor compared to the other settings. And also, a small runtime leads to less pronounced results. We then estimated the growth parameters and their uncertainties in all regions. We also estimated the total mortality distributions using the growth parameter distributions as an input. As the mesopelagic species are currently unexploited, we assumed the total mortality to be equal to the natural mortality. We presented the uncertainty of the parameter estimates as the difference between the upper and lower limit of the 95% confidence intervals. We also estimated the selectivity parameters L50 and L75 and their uncertainties, indicating the length at which the fish have 50% and 75% probability to be caught. The estimated selectivity ogives reflect the differences in selectivity in the regions, with high probability of being caught at small sizes in the Norwegian waters, for example, where the small meshed microzooplankton nets were used. The estimated parameters can be used in further analysis using TropFishR, for example to estimate exploitation rates or for yield per recruit modeling. In yield per recruit modeling, biological reference points and stock status can be estimated by predicting the theoretical yield and biomass per recruit for a range of fishing mortality and gear selectivity assumptions. This method assumes von Bertalanffy growth and equilibrium conditions and requires length frequency data, the estimated growth and mortality parameters, selectivity information, and parameters of the length-weight relationship A and B. If maturity information is available, reference points related to the spawning potential ratio can also be estimated. Now these are just 2 examples of other functionality in the TropFishR package, but there is much more. Further information, a reference manual and several vignettes can be found through CRAN, and more information about the methods and the package can also be found in the FAO manual and several published articles.